I'd like to tell you today about how NeoGenesis in our laboratory processes our stem cells to make our molecules for the hair and skincare products. This is very important. There's very distinct features here involved in our processing versus the way other companies do this. First of all, you know we use 2 or more stem cell types that we stimulate in the laboratory, allowing the stem cell types to fully process the proteins and other molecules so that the proteins, for example, exist in 3-dimensional states. And if you don't allow the stem cell to fully process those proteins, they won't have all the complete amino acid sequences, they won't fold up properly. Beyond that, we also allow the stem cells to fully process the molecules, the proteins, package them into the exosome. That's the liposome-like nanoparticle that protects the molecules and then allows the molecules to perfuse through the tissue through barrier function and target neighboring cells. And so our technology in the laboratory allows all of this processing to go on so the proteins and other molecules have the full sequence, fully formed, 3-dimensional configuration is correct, packaged into the exosomes, and then released. So that it is a very natural processing that mimics the way Mother Nature processes these molecules in our body. That's very different from some of the other companies. Some of the other companies, of course, use only one stem cell type. They don't mimic Mother Nature. They don't allow the stem cells to fully process the molecules. They'll actually do an extraction, a very harsh, old-fashioned extraction of the proteins from the cell using chemical techniques that actually can deform, in and of itself, that chemical deforms the proteins. And secondly, since the protein isn't fully formed by the stem cell, it's not going to fold right. It's not going to have the right sequence. It's not going to fold correctly. And then it's not going to be packaged into the exosome that protects it and enables the molecules to be delivered to neighboring cells. So that technology is an older, it's a simpler, it's a less expensive technology than what we use with our fully formed, fully packaged exosome technology at NeoGenesis.